Okay, so yesterday I presented on some of the natural history from our newborn screening population, and I really appreciate the feedback. I had a great conversation with the Krieger group last night. I found it really helpful, so I had some really helped crystallize some ideas for me. Today I'm going to talk about the Cerebral ALD mouse model that a couple of the scientists in my lab developed. We'll talk about how we might use it. I'll try and keep it a little bit as much out of the weeds as I can. So relevant disclosure. We are starting to, and our hope is actually to do work with companies to screen their molecules in the model here. So there's that internal, that inherent conflict of interest. But to date, we're really just kind of working. I'll show you the data that we have, with just no industry work involved at this point. I want to point out Azad Hashemi is the senior scientist at my lab who really conceptualized and built the model. I want to credit Josh Bonkowski with some of the key ideas, and then Isha Srivastava had done some of the human histology work shown here. Okay, so we'll jump right into what is cerebral demyelinating ALD. Here's just some of the ALD background, just to give you a sense of where we're coming from here. I guess the thing I want to point out at the beginning here is we have a pretty limited understanding, and this is true of most disorders. This isn't unusual for ALD, but we have a pretty limited understanding. We know where things start. We know they start with the ALD gene, ABCD1 mutations. We know what that causes. It causes a specific protein change and a buildup of a set of fatty acids. The area from there onward is a little fuzzier, and so this is an area that we're hoping new model systems can help evaluate. But here's what we know. So we know there's kind of a bimodal distribution for cerebral ALD. So young boys, as people in this room know, are kind of highest risk, and then younger men, looks like in their 30s in particular, are at relatively higher risk as well. But the distribution of risk is across the lifespan for the most part. So I don't mean to say that there are only certain ages at risk. It's just that we were talking yesterday about chance of rain. Chance of rain is highest during certain periods of life for cerebral ALD. And there are some people that do not get cerebral ALD, and that's a story we won't cover here. But there are a few key locations. We know how the lesions evolve. Typically, once they start, they keep going, and that's unique compared to other inflammatory brain conditions that start, stop, and sometimes never start again, or start, stop, as is the case of multiple sclerosis. We understand a bit of the histology. This is where things really started for cerebral ALD. There were some pathologists who described the first cases. There's a lot here that looks like multiple sclerosis and other diseases, at least superficially. And so there are some similarities to be had there. They are not completely similar diseases, obviously, but some similarities. And this is the concept of the black box. This is a term that we use to describe a system where things happen that we cannot see. And this is how lots of diseases are to people who try to understand them. And so we know kind of what goes in, we know something happens, and then we see what comes out. In other words, you have a biochemical change and a genetic change that goes in, something happens, and what comes out is brain inflammation. And a lot of science has really focused on these black boxes, what's happening inside this wall. And this is what scientists are really motivated by. This is what is fun for scientists, is to solve these problems. Lots of people have contributed to the knowledge, to progress. At each stage, we're standing on the just the next level of what is known, and I certainly say that's the case here. For cerebral ALD, it's been a steady contribution for 100 plus years. We have a number of model systems we mostly talk about an ALD mouse. There are other systems that people use, and there are new systems. I don't think they have, so I guess it is on here, but there are tiny artificial brains that people are making that are quite interesting. There are new genetic models. I know other labs are working. I think there are some. Hopefully, we'll hear more about new models in the year or few ahead. But the mouse model has always, because the mouse model drives so much of what happens in biological science, often for good and sometimes just with lots of limitations, or I should say always with lots of limitations. It's disappointed in ALD in that it does not spontaneously develop cerebral lesions. One caveat here is I would say that is also possibly one way to think about humans, is that humans with the ALD gene change don't spontaneously develop. There's something else that happens, and we know this, like, for example, from identical twins, where one gets something and the other gets cerebral disease and the other doesn't. So something is happening in one person that's not happening in the other. Is, again, nothing that is terribly unique to ALD. This is really true of just about any biological process. There are usually interfering or we often call them environmental factors, but sometimes we use that term environmental factor to cover all the things we don't understand, and I'd say maybe that's the case here too, but there are things happening. Our lives are busy, they're complicated, and there are things happening in our lives, in our bodies that we're not privy to, and so understanding those things is an ongoing process. What we think happens in humans is we have, start with a mutation, we have higher fatty acids. We think, we're all comfortable with the idea that oxidative stress is part of a process, whether it is necessary and sufficient, or whether it is directly involved, issue of debate. We think so. Inflammation is clearly part of what is happening in these brain lesions. There are lots of immune cells there. There's lots of damage happening. How these things start and perpetuate is really the question. And why they don't start and perpetuate in the mouse is another question. But this is what we sort of set out to look at. These are a few of the kind of, this is a little bit more in the weeds than we'll need to get here, but this is some of the kind of, for us, these were some of the check boxes, like things that we wanted to see in the mouse model that would be useful. There are other things here that we can delve into that so far we don't exactly see in the mouse model, and those are important limitations for any model system, but or things we haven't looked at yet, things like that. But these are a list of things that, this is what we would hope to see in a model that we do see in human disease. Josh was working on a fish model. Josh Bonkowski is a scientist at University of Utah. He was working on a fish model, and he felt like he got something to work. And he said, "Why don't you try something like this in the mouse?" And we sort of set something up that we thought was similar, and it worked on the first try. It was really nice, and it was partly because Dr. Azad Hashemi was a really experienced postdoc who'd worked in an MS lab and had used a lot of the MS models, and she said, "I know when the timing happens for these important things, let's match them up here." And so what we did was we combined essentially two MS models. One is a cuprazone intoxication, which is an oxidative stressor. It's basically oxidative stressor you put in the diet. It's a copper-based stressor. The mice just eat it as part of their regular food, and it causes demyelination. So it's artificial. This doesn't really happen in humans in the exact same way, but it's one of the models we use for MS for studying this. Usually it's very mild demyelination. It self-resolves even if you continue the diet. The EAE model is commonly used. It's basically like immunizing. If you took your own brain proteins, your brain myelin proteins, and tried to make yourself respond to them like an allergy, it's called experimental allergic encephalomyelitis. So that is with the model, and you basically use a brain protein to try and make, and some inflammatory things to try and make the mouse and have an immune response in the brain. Now, these two models normally, they don't often go together, but they have been put together before. They've not been put together in ALD. That was the innovation here. There are limitations of these two systems. Cuprizone and EAE in and of themselves don't cause significant brain inflammation. Cuprizone doesn't cause much inflammation, and EAE doesn't cause really any brain inflammation. It causes spinal cord inflammation. We wanted to try and find brain inflammation, and so that's what we were set out to do here. We got very good results right at the outset, and this has been consistently replicated, at least within our lab. And so what we see here is basically on the y-axis is the severity, like how severe the symptoms are in the mouse in terms of weakness in the limbs, like tail and the legs. The symptoms were more severe in the ALD mice who got this combination treatment, and everything else, including the wild-type mice, were a step below that. When we looked at the brains, so top here is wild-type. There's still a lot of inflammation in the wild-type mice and in ALD mice. Some markers of inflammation were very similar. Others were clearly more robust in the ALD mouse. And so the actual, like this is just a macrophage activation marker. That was pretty similar between the wild type and ALD mouse when we induced it. But other markers were significantly more significant. We had more demyelination in the ALD mouse. We were able to get a nice MRI correlate in the ALD mouse, which we thought was helpful for people who want to. It's relevant for some translational studies. We think that's a useful feature. We were able to get gadolinium enhancement in the mouse, and so these were all really useful things that really even for most of the MS models don't do this, and so we were excited to be able to get these to work. We really did see that blood-brain barrier, markers of blood-brain barrier breakdown were much higher in the ALD mouse in particular. Like that was really that seemed quite different to us, that there were higher markers of blood-brain barrier breakdown on the MRI and on the staining. So this is a fibrin stain. Fibrin is basically a component of blood that helps with clotting and other things. And when the blood-brain barrier breaks down, it deposits in the brain and causes its own form of inflammation. That was much, much higher in the ALD mouse than in the wild type. We saw many more spots of inflammation in the ALD mouse. So these markers these are like the around each blood vessel. When it leaks, it lets immune cells into the surrounding tissue. This is characteristic in ALD, or it's present in ALD, and we found it at very high, much higher levels in the ALD mouse compared to wild type. So there were areas that were really much more highly inflamed. We found different types of immune cells we were hoping to see. So we saw these macrophages are the cells we think right now the field, we and others in the field think that macrophages are these kind of cells that are present in the brain as microglia. They're highly involved in the process to begin with, and then they send out signals that bring in more immune cells like called T cells and B cells and other cells. And these are not present also in most MS models either, and so we were excited to see them in our ALD model because that is present in real ALD tissue. So we're getting a wide spectrum of things that we think replicate disease. We saw really high levels of oxidative stress in our ALD mouse, much, much higher than in the wild type. We think this is a promising marker we do hope to investigate further. And then this was one of the areas we did a few crossover human studies with as well, and this is it's called interleukin 18. It's a marker, or can be associated with a marker of a certain kind of cell death. So it gives you a kind of a specific target aim for in therapy. We saw high levels of this in ALD human tissue and then ALD mouse tissue. So what we found at the end, and this is a little bit self-fulfilling because there are things that we can talk about that don't replicate here, but what we found is actually really, we thought, we were very pleased with ourselves. We thought this is a really good list of things that we think this model can do that we haven't been able to do before. From here, what you want to do is there's always more questions to ask about how good the model is, what are the key limitations that might prevent you from studying one area versus another. How do you work around that? But of course, from here we want to say, well, let's start and see where we can go next in terms of understanding disease pathogenesis and then new treatments. Our lab, as you all know, we've worked on vitamin D trials in the past. And spoiler, our phase 2 vitamin D study is not funded, so we probably will not be doing that in the near future. We'll be updating you with other strategies. But we wanted to look. So what does vitamin D supplementation do in the mouse? And this is really a prevention model. So in other words, if you take vitamin D early in life, does it reduce the chance of rain? Can you reduce the chance of rain by getting sufficient vitamin D? This doesn't literally answer that question, right? It's a mouse model. But what we did was we put the mice on, divided the mice into 3 groups and gave each group different levels of vitamin D. One made them very insufficient, one gave them sort of normal levels, and then one gave them higher levels. And the insufficient ones had significantly more, or significantly lower vitamin D levels. The medium and higher mice had similar vitamin D levels. This is just their vitamin D levels here. And then when we look at, so higher is worse here. So these, here the lowest mice had pretty similar just disease severity in terms of symptoms compared to the medium dose, and the higher dose had slightly better clinical severity. Clinical severity is a very rough measure here, but we do use it. The immune infiltration seemed to be different in the 3 groups, meaning the more vitamin D they got, the lower the immune infiltration was. So here, lower is still better. So this was our high dose. The effect was true in wild type. We ran this in wild type as well. So it was not, this part was not different. We saw lower levels of demyelination, which would kind of go along with the lower level of inflammation, and then lower levels of oxidative stress. And again, all of these kind of correlate with the level of inflammation you see. Now, good, we can see this. This is helpful. This is also, by the way, this has been done in various forms in MS mouse models many times. The vitamin D has been studied there. So this wasn't entirely novel, but it was a useful proof of concept for us. What we are aiming to do now is understand more of the mechanism for vitamin D and then for just ALD, cerebral ALD in general, and do more crossover studies of this is what we see in the mouse, does it bear out in the human, and then can we apply therapies to fix these things. So this is really, I would say, where we are now, and that we have, our conceptual model here is humans and mice with an ABCD1 gene defect are not fated to develop cerebral ALD at the outset. Something else happens, we don't know what that something is, but in humans it happens in the background without our awareness. In mice, we try to induce something that we think is similar. So we use an oxidative stressor, cuprazone, and we use an immune stressor, this EAE, it's basically immunizing mice to their own brain proteins. And we are able to produce something that's quite similar. So in other words, lack of a model has limited us. So there are limits what questions we could ask before. We now hope we are in an era where we can ask new questions and answer questions, and specifically answer questions that are relevant to developing new treatments. So that is how we want to use it. we hope that this will help be one of the things that helps accelerate and encourage and help companies. Essentially, if you're a company that has a drug and you say, gosh, this might work in your clinical disease, how do we know if it's not a gene therapy, say, or even if it is a gene therapy, but let's just say you want to know, you want to have some de-risking. Companies are typically driven by investors. The people in the company often care very deeply about who they're helping. They want to help people. They're trying to do good. The investors might be less concerned about that. They want the company to make money. It's sort of a third party here that's driving that process. The investors and the company both want to succeed, but the company is potentially accountable to the investors. They need to show due diligence that they've done all the math to suggest that their product is going to work. This we hope will help. So we are talking with a couple of companies to try and figure out if there's a way to do this. We hope we're on our way in that direction, and we have some working on some exciting new grants and opportunities. We did get funding from ELA to study a novel antibody targeting fibrin, one of these blood proteins I mentioned. So we're very excited to begin working on that. So I'm going to end there and take some questions if there are any. Thanks.
So thank you for that. I'm just curious, how do you create the oxidative stress in the mouse model? I saw there was a drug involved, but how similar is that to the real world?
Yeah. So the drug involved is not something that would be present in anybody's diet. It's a molecule called cuprizone. It's a copper-based molecule that gets incorporated in the cell and induces oxidative stress within the cell. So that is not happening, and I know none of our kids are eating cuprizone. It's not present in the real world. Are there other things that can cause oxidative stress? Well, yes, I had a drink with Ollie last night. Alcohol is an oxidative stressor. Thank you, Ollie, very much. There are lots of other stressors in the real world that can contribute to that. But suffice to say, we don't. If we really thought there was something, by the way, I do recommend for men in particular. I do recommend alcohol in moderation, so not too much. But there are really things that we could advise avoiding, we would. We don't really know what they are at this point. Did you have a question? Okay.
Thanks, Keith, and your gentle persistence has paid off. So congrats on that cerebral ALD model. So needed. And as I think about this, as much as it's an important new milestone, it brings also risks. Some of the hopes that you have proof of concept for future treatments, but also potential misreadings of what could be efficacious in humans. So to understand the limitations of the model is probably as important as understanding what it models, because we wouldn't want to withhold something in human that is not panning out in this mouse because it maybe is not replicating all aspects of cerebral ALD, right? So you beautifully outlined all the different cells that are affected, but there's a very specific, as you know, zonal pathology, propagating pathology, and all of those things, come together in human, and the mouse might be replicating parts of it, but not everything. And how can you parse that out, and how can you guide us to use this model in the right way, not the wrong way? Yeah,
so I would actually qualify that even further and say, I promise you it's not replicating human disease perfectly. But in the sense that it's a model. So just to broadly speak, when we speak of model systems, they are always limited. They are, like, if it was perfectly mashed, it wouldn't be a model, it'd be the real thing. And so it is one of these things, but understanding where the model misleads or fails is really key. And I will add that is I would say it's a, right, it's like a double-edged sword. You have something that you are excited about and think will work, but you don't know what you don't know yet, and so you have blind spots. We don't know what the blind spots are really yet. So the question, and this has been I'll tell you that for the MS field and many other fields, they cure everything in the mice, and then they get it to a trial, and it disappoints. Now, this always depends a little bit on how close the model is to the real system. For Alzheimer's, for example, my opinion is that Alzheimer's field is potentially the biggest failure of science in modern times. And the reasons for that are many, and not probably totally relevant here, but one of them is probably the way you think of the disease and the way you model it. We do have an advantage here that we can start with what we know is the foundational kind of ground truth. We know the gene. That helps, but then everything else becomes fuzzy too. So I can tell you more concretely what we are looking at to try and match this. There is some new technology looking at kind of like, you basically, just like you look at brain tissue on a slide, you can do this to an extent using kind of molecular sequencing. You can do this with lipids, you can do it with RNA, we call spatial transcriptomics, and you can do this with lots of other techniques. That's one of the areas that we're looking at starting. It's qualitative at this point, so we're saying, all right, what are the pathways that we think we see in human disease? And we don't have that data really yet, and we're excited to get that. Isha Srivastava is doing that, and then Azat Hashemi is working on that same in the mouse. We have some of that data already, and that is a start, but that's at least our first step in getting there. That's all unfunded work. Our next step is really to get we hope funding from if the NIH, if it still exists, in the next few years, and other agencies to try and fund these things to figure this out. So I will pitch in here too. This is where, for those of you who are watching, scientific systems teeter in here. This really is where scientists can get things to this stage, and then things from here, like we really depend on particularly NIH funding to get projects beyond these stages. So I would describe this as actually preliminary data for what we need to do next, and the next is really a bigger investment. So go ahead.
Follow up?
Yeah.
Just speculate briefly on the fact that you need a two-hit system. Many of us have done EAE in the past, Klaus Nabe's group, my own group, Celia Kassmann, and us, we've compared. And the ALD mouse has always been more mildly affected with EAE alone compared to wild type. And it's great you went to the two-hit, but what is it telling you, and what is the fact that EAE alone may not suffice, and how do you think of the mouse, at least pathophysiology in that respect?
Yeah, well, part of it is understanding the limitations of the original models, right? So EAE itself does not induce brain inflammation in the wild type mouse. It induces spinal cord inflammation in the wild type mouse. And so it wasn't, in other words, it's not a good for whatever reason, and it's not really what it does. To walk through very coarsely how we're thinking about it is the oxidative stress for Cooper's-Hone E, we know, induces inflammation in the myelin in the brain. So it primes the system to be at risk. Something else happens, the inflammation happens afterward, and it creates this, you basically, you light a match and then you pour on the fuel. Something like that. But why that is needed here, I don't know. I mean, it's actually needed for wild type systems too. So in other words, this produces something similar in wild type mice. Here it is just more severe. So one other way to look at this is ABCD1 deficiency makes the MS model much more severe. And I think it's fair to say much more severe, not in every aspect, but so that is one way to think of it. So in other words, we can see that the ABCD1 deficiency is doing something unique to predispose to brain inflammation. I don't know why we need this second hit. I don't know. I mean, this is just a problem. It is a problem with the system. You know, the more artificial things you add, the more further it can get from the truth. Go ahead.
Yeah, actually, that's a very good point. It's like you could use a little more. If you dived in a little deeper, you could maybe find some clues there. And I'm wondering about that because, Josh had done the work with the zebrafish where he showed the differentiation defect in oligoprogenitors.
Yeah.
My understanding from an experiment that Aurora Pujol did on cuprizone only, so cuprizone induction in ABCD1 knockout, was that she saw the same thing, that differentiation of oligos. So everything looked the same between the cuprizone ABCD1 and cuprizone wild type, except the oligodendrocyte differentiation was slowed down more. When they were remyelinating, so the remyelination phase was slowed down, which would go along with Josh's findings. Did you look at that in this? Because you could probably go back and still look at the specimens you have and compare the two with each other.
Yeah, we have a Stanford undergrad who's working in our lab trying to study oligodendrocyte differentiation in the model, all I can see so far is we haven't found a good mark. We haven't found a good marker.
Michelle, she's right.
What's that?
Michelle Mongi, she's right there. She can.
Yeah. I mean, we haven't found the marker that differentiates them. I mean, we have markers, but we haven't found one that kind of differentiates the one with the yeah.
Just out of curiosity, with this model, have you thought about doing things to the adrenal?
Thanks for
and
asking.
Just block, they're drugs that you could just block cortisol production and see if that changes anything.
Yes. So I was Troy. I don't know if he published it or he showed data on elevated ACTH in the which I thought was totally interesting. And yes, those assays are right now we need funding to look more, but I think it's a really, really important piece here. One, we need more endocrinologists in our group meetings to ask us questions like that, because we're all the one-track mind. But no, we haven't looked. Is the short answer, Molly, and I think it'd be really interesting to see what's happening to the adrenal, just ACTH production in the mice under stress.
Not only just looking at it, but also have
the tissue pathology.
Looking at the tissue pathology, but you could model, even if the mice aren't getting adrenal insufficiency fast enough, you could model giving them adrenal insufficiency to see. I recognize time is. I recognize time is of the essence with all of this, but you could model those with adrenal insufficiency, those without, those getting replacement with adrenal insufficiency also.
Yeah, that's a really good point that we haven't considered in terms of trying to adding adrenal insufficiency to the model. So maybe we'll pick your brain on ways to do that. I like that idea. Okay.